Thank you very much. My pleasure being here. I'm not, and our group are not neuroscientists, so we are developing new imaging modalities, especially for brain imaging. So we are kind of bioengineers or even engineers, I would say. So excuse me for a little bit technical talk, even though I will mainly show images today. No formulas, I promise. So our group is doing a lot of imaging-related things. What I'm going to talk today is mainly about optical developments, microscopy, optoacoustic imaging. But we are also engaged a lot in ultrasound, functional ultrasound, MRI, and other methods, especially the combinations between different modalities, and more recently also on applying ultrasound to brain therapy and monitoring the procedures. I'm not going to touch upon this point today. So I'm going to talk about mainly the brain, and as you know, intravital imaging or in vivo imaging is very important for studying the brain, and that's how one of the main goals of also the brain initiatives across the world were formulated, trying to develop a new set of technologies to monitor and manipulate activity of millions of neurons, both individually and collectively. And to do that, ideally, you need to have very high resolution. You need to be able to record fast activity, ideally related to, for example, voltage changes or calcium changes. You want to do it on a whole brain level and also in living and intact organism. And there's no imaging modality that can do all of it, at least not in mammalian brains so far. Nevertheless, there has been tremendous progress over the last decade, I would say. And so let's see what is there, at least for some of those aspects. So if you want to image the whole brain, that is possible today, and there is technique that can dissect the brain without, like virtually dissect the brain. So tissue clearing, right? So you can clear the brain. You can even clear the entire mouse. So we have a big initiative in Zurich called Mesospeem of using light sheet microscopy to really visualize different cell populations in the brain and so forth. And we use it. We use it for many purposes. So one of them is shown here. So this is a brain of Alzheimer's disease mouse, where we could label, by injecting in vivo a contrast agent, we could label all the amyloid beta deposits in the brain and visualize it in 3D with very high resolution of a few microns across the entire brain. Obviously, this is very invasive, right? So you need to clear the brain, which could take a few days up to a week. And the main disadvantage, this is just a snapshot of one time point, right? This is a dead sample. So this is actually analogous to what people have been doing for centuries, basically those anatomical dissections, so since the ancient Greek times. And I always show the slides because of this. So there's a picture of the anatomical theater here in this building. So I was really eager to visit it, and I managed to squeeze in between the school tours with the kids. So they let me in, kicked me out after a few minutes, but I managed to actually stand there and see this because I show it in my molecular imaging course every year. So I was really happy to be able to visit it. So this is all about ex vivo, right? So you can nowadays study individual cells or subcell structures. What is out there for really in vivo functional brain imaging? So the workhorse is obviously fMRI. We had a lecture today. Nowadays, with techniques like opto-fMRI, you can stimulate different cell populations in the brain. So with optogenetics, for example, so you can selectively stimulate the dopaminergic system and visualize the bolt signal across the entire brain. The penetration is whole brain, also for humans. But of course, bold fMRI is limited by the spatial resolution. You cannot see single cells, or you cannot even relate the signals directly to a neural signaling. So it's like indirect hemodynamic readings. And temporal resolution is also poor, right? So you could maybe read out 1 volume per second or something like that. There is another technique which becomes very popular, functional ultrasound, or the more advanced version of it is, or the more recent one is ultrasound localization microscopy, or ULM. So with ultrasound, you're much faster, so you can capture the entire brain in 3D and image it very fast. But you're still limited by the hemodynamic readings. So ultrasound reads out blood flow mainly, right? With ULM, you can go down to single capillaries even, so you can break through the resolution limit and characterize flow, also quantify blood velocity, for example. But you're still reading out the blood-related signaling and not directly the neurons. Now, if you want to do it through the skull, that's another issue. And again, you are on the macroscopic level, right? So mainly seeing the vessels, not the cells. If you go down to cells, that's a different scale. So there are many techniques that can do it in vivo as well, like two-photon microscopy. So this is an example of a miniature two-photon microscope attached to a mouse brain. Obviously, you need to open the cranial window, install a glass window there. It's a chronic implantation, right? Nevertheless, you are now on a single cell level and you can visualize small populations. The field of view is not great, so you're not looking at the whole brain. So maybe a few hundred neurons at a time, a single two-dimensional slice. To do it in 3D is quite difficult. So again, you're in different scale, but your field of view is now very small and it's super invasive, right? So it's craniotomy, so you can actually disrupt some vascular structures in this way. So I'll try to explain you in the next slides how we try to improve what is available in terms of intravital microscopy. And the first technique that we developed a few years ago is called high-speed large-field multifocal illumination microscopy, or LMI in short. And the idea is to split your beam into multiple mini beams. So there's a pattern of tiny beams that is going through the skull. This is a fully transcranial technique now, so skull is intact. Sometimes we can also do it through the scalp, through the skin, so it's fully non-invasive. And it can cover the entire cortex of a mouse, okay, so 12 by 12 millimeters. Now what we do with this beam pattern, we scan it with acousto-optic modulator very fast. So within a few milliseconds, we can form an image of the entire cortical vascular tree. So the spatial resolution is very high. The temporal resolution is also very high. So what you get at the end is this kind of image here on the left, done completely non-invasively. Okay, so we can visualize individual capillaries through an intact mouse skull over the entire cortical area. So the previous technique that was used for this purpose is called laser speckle imaging. Here you can see it on the right. Not sure I can show it with a pointer, no, but you can appreciate the improvement in the spatial resolution. So we are really looking now at single capillaries, and we can visualize the perfusion in the mouse brain non-invasively. And now we can start studying, for example, how the collateral flow is compensating for the loss of blood perfusion in stroke through those bypass collaterals, and this is all now visible also non-invasively. Now, if you apply to a model, to a disease model, for example, stroke, now you can characterize the perfusion deficits in stroke very quantitatively and with high precision. So we could develop different types of indices, for example, to see how the early recanalization is shown to effectively save the penumbra. So now the penumbral area, we developed this new index, which is called blood flow index, that characterizes the amount of blood supplied to different areas of the brain. And it is much more accurate than the previous techniques that were developed for this purpose. So with LMI, you can do other things. So this is a fluorescence-based method, right? So you need a fluorescent dye to make it work. So if you make your dye targeted, for example, to amyloid beta plaques, you can now start visualizing the individual plaques through the intact skull of a mouse, and you can do it longitudinally then, right? So you can see really how the plaque load in the cortex is developing over time, because you don't need to remove the skull anymore, and you don't disrupt the vasculature under the skull. So you inject the targeted contrast agent, and after 2 hours, you can look at the plaque distribution in the cortical areas of the brain at a resolution of about 7 to 10 microns. So you really can characterize the size of the plaques, the distribution of the plaques in the cortex, and so forth. So here, those green dots, they are individual amyloid beta plaques. Another technique we developed for a similar purpose is called wide field fluorescence localization microscopy. And the idea of this technique is actually similar to super resolution ultrasound or super resolution microscopy. So what we do, we inject particles, microparticles usually, that are fluorescent, and we let them flow in the bloodstream. And the next step is to track all those particles as they flow on a particle by particle basis. Okay, so we track each individual particle, and you see a picture that we get with the camera. Now, after localizing and tracking all those particles for a few minutes, we can reconstruct an image like this. Okay, so this is even higher resolution than the previous technique, and it's again done transcranially. Okay, so this is really a super resolution technique. If you try to look at the brain with some fluorescence particles with just planar fluorescence technique, everything will be blurred, right? Because you go through the skin, through the skull, everything is smeared. But now we are able to track the individual particles much more precisely than just looking with a simple camera approach. Now, this technique is also quantitative in terms of quantifying the blood flow, because we track particles that flow in the blood. So the tracking means that you read out the blood velocity. You get it for free, right? So basically with this technique, and we call it diffusion optical localization imaging, or DOLI in short. So with this technique, we are not only able to reconstruct with high precision the cortical microvasculature, but also quantify the blood velocity and the flow direction, because we track all those particles. Okay, and again, compared to wide field fluorescence or laser speckle imaging, the performance is much better in terms of resolution and also quantification. Now, since we have the camera and we have the DOLI approach, we can quantify both the blood flow and, for example, calcium changes in the brain. So if the mouse expresses, for example, genetically encoded calcium indicator, we can read out simultaneously the calcium changes in the neurons and the blood flow changes to relate to, for example, neurovascular coupling in the brain, right? So actually, this is an improved version of technique where instead of injecting fluorescent particles, we manage to label red blood cells. So we draw a little bit of blood out of the mouse, we label it with DID dye, and we re-inject it into the blood. And then we track actually red blood cells, labeled red blood cells instead of particles, which are more toxic in this case. And here you see on the right some movies of stimulating the mouse, the hindpaw of the mouse with electric current, and we see how, or for example, whiskers, and we see how the blood flow changes on the individual capillary levels together with the calcium changes in the brain at the same time. And we again applied this technique to stroke. So we looked at the vascular responses to arterial occlusion. And with the DOLI technique, we are now able to really characterize the flow direction in individual and capillaries and the velocity of the blood flow, how it slows down and restores after treatment, for example, and so forth, with high precision, again, on the level of 5 to 7 micrometers through an intact skull. Okay, so that's it for all those microscopic techniques. Obviously, the limitation of it is the depth, right? So what I was showing you, we overcame the transcranial problem of intravital microscopy, but we still didn't overcome the depth. problem, right? So all those techniques can be done up to maybe the half of the cortical depth, okay? What would you do if you want to go deeper? And this is where hybrid techniques like optoacoustics come into play. So we developed this technique called multispectral optoacoustic tomography, or MSOT in short, where we use short pulses of light for interrogating the brain. But what we read out is an ultrasound response to light, okay? So this is a combined technique, or we call it hybrid technique. That's why it's called optoacoustic. So basically, the imaging contrast is still optical because we use light, but the readout is ultrasonic. So we are able to overcome the light diffusion in this way. So if you look up optoacoustic imaging or photoacoustic imaging, you would usually see those beautiful images of vasculature. So here in the center, this is a movie of stimulating the mouse brain with electric current again. So as you run the current through the pole, you would see that the oxygenation in the somatosensory cortex is changing. Now this is done label-free because optoacoustics is very good in visualizing blood vessels and blood in general, and also blood oxygenation. So this readout we get for free in optoacoustics. You don't need to inject any contrast agent because this is a spectroscopic technique which is similar to a pulse oximeter that you put on your finger, but it also has a very high resolution in resolving those blood oxygenation changes. And on the right side, you see a movie of oxygenation of placenta that was taken longitudinally, okay? So this particular optoacoustic technique is called scanning microscopy. So light beam is focused in this case, and you scan it similar to photon microscopy, okay? So the depth is still not great. You only see the cortical areas in the case of the brain, but this is a fully label-free technique, and the field of view is very large. Of course, if you want to go deeper into the brain with optoacoustics, you cannot focus light anymore. And this is the technique that we developed for deep tissue imaging, which is called MSOT, or multispectral optoacoustic tomography. And in this case, we just shine light, usually near-infrared light, that scatters around the tissues, and we capture for every pulse of light three-dimensional responses from the tissue using ultrasound transducers. And to implement it, you need a powerful laser, which is quite expensive, and some readout electronics, which is also not cheap, and this kind of spherical array transducer. I'm not going to go deep into explaining how it works, but just will show you the result. So this type of approach is very fast, first of all. So for every pulse of light, we are able to read out a three-dimensional picture of the entire tissue volume. So we can easily cover the entire mouse brain, in this case, with resolution which is ultrasonic. So it's not microscopic, I would say, but on the order of 100 to 150 micrometers. So in this movie, you see actually a four-dimensional data set acquired from a human breast. So it's up to a depth of about 2 centimeters, okay? So the technique can go very deep into the tissue. It's very fast. It can acquire the blood vessel picture very precisely with fairly high resolution. Now, since this is a whole brain approach at least for rodent studies, and it's totally non-invasive again, so skull is intact, skin is intact, we started off with studying hemodynamic changes in the brain. And in this particular example, what we did, we induced epileptic seizures in the brain, and we tracked how the seizure propagates through the brain in 3D. Now you inject some epileptic agent, and you wait till the seizure starts, and then we can read out how it propagates, starting from the cortex, going into the thalamus. So it's kind of cortical depression wave that is going through the entire brain, going into the thalamus, and ending a loop in the cortex, actually. Technique is very fast, so we can read out 100 volumes per second, so it's enough to read out hemodynamic changes, okay? Now, this is powerful, but if you want to go into calcium imaging, for example, you can also combine optoacoustics with just planar fluorescence imaging, and this is what we implemented in this work, where we inserted a fiberscope through a hole in the spherical array transducer, so we are able simultaneously to read out fluorescence calcium indicator changes in the brain together with the hemodynamic changes. So now we can link the calcium changes, for example, in response to a stimulus, together with the hemodynamic changes, for example, oxy and deoxyhemoglobin, which is read out by the optoacoustic technique. And maybe something even cooler, what if you combine optoacoustics with MRI? So there was a previous talk about fMRI, and there is, I think by Lorenzo Pini, right? So, and he was talking about bold signal in fMRI, and this is actually quite controversial what it corresponds to, right? For example, many people explain it as changes in deoxyhemoglobin, and we saw that's not really the case, so I'm going to show you some data about it. So we implemented a hybrid magnetic resonance optoacoustic tomography device, so we actually inserted the spherical array transducer into the MR bore, and in this way we could do functional optoacoustic together with functional MRI, okay? And the results were actually stunning, because once we did it, now we start stimulating the mouse with electric current, for example, and we read out bold signal with fMRI together with oxy and deoxy changes separately, actually, read out by the optoacoustic technique. And what we saw, and this is same temporal scale, same spatial scale, so we capture the entire brain with similar resolution in temporal and spatial. So what we saw actually is that the temporal shape of the bold responses, it better correlates to the total hemoglobin, which is basically the blood volume. And it has little to do with deoxyhemoglobin changes in the brain, okay? And this was also confirmed with resting state functional connectivity studies, because we can do this simultaneously again by fMRI and by optoacoustics. And again, we consistently saw that the resting state changes read with optoacoustic, so the resting state fMRI corresponded better to the total hemoglobin changes, not the deoxyhemoglobin changes. Which puts under question, you know, even the acronym of BOLD, because BOLD is blood oxygen level dependent, right? But if your signal doesn't correspond to deoxyhemoglobin, but just to the blood volume, how would you interpret oxygen changes, right? So that's the question. And this was actually strengthened by a follow-up study where we implemented another device where that records fluorescence calcium changes together with BOLD. So we inserted a camera or a fiber bundle, a fiber scope, into the MR bore, and we split the fluorescence readouts into two channels. So one is could read, for example, astrocyte signals with RCamp, and the other channel could read the GCamp, okay, the neuronal signaling. And all this is done in fMRI, okay? So we read out also the BOLD changes from the brain. And what we clearly saw, so we did an interesting experiment. So we induced hypercapnia, okay? So we let the mouse breathe SO2. It's my last slide, actually. So we let the mouse breathe CO2, and basically we saw that the BOLD responses completely vanish at 5% CO2, which is quite expected, right? So the CO2 is supposed to inhibit the BOLD, right? But then we saw that actually GCamp and RCamp signaling is preserved. So basically there is neural activity and also glial activity, right, astrocyte activity, but no BOLD signal, right? So the neurovascular coupling is completely vanished, right, in this case. So there was some sort of disruption of astrocyte to vessel signaling in this case, okay? And this could, we could only detect it because we had a hybrid device, right? So we read out both the astrocyte signaling and also the vessel signaling from the BOLD, right? Previously, it was done separately, so you could not do it simultaneously. So I'm not saying that, you know, my point is not that bold is not a useful approach. It's actually very useful, and I started liking when we started doing it even more. But you have to be careful what you interpret by reading out bold. Okay, so our assumption now is that it's very difficult to predict, for example, oxygenation changes in the brain with bold. So the acronym itself is a little bit misleading because you think that you read out oxygenation, but what you actually read out is, and it's based on many of the papers that we published in the last years, what you mainly read out is the blood volume, okay? So you have to be careful, interpret this as oxygenation changes. Of course, it depends on your experiment. So in some experiments, bold signal would still somehow correspond to oxygenation changes, but in many other cases, it will not, okay? So you have to be careful how you interpret this. Okay, so that was it for today. Of course, I would like to thank my group of very talented, and all the credit, by the way, goes to them, not to me. I'm not even doing most of those experiments anymore, unfortunately. Yeah. And thank you very much for your attention.